Not all enzyme regulation happens at the active site. Many enzymes are controlled through allosteric regulation, where molecules bind somewhere else on the protein to fine-tune its activity. Let's break down what this means and then look at cooperativity as an example. The word allosteric comes from the Greek word allo, meaning other, and steric, meaning shape, so it literally means other shape. These enzymes have a binding site separate from the active site. When a regulator binds there, it changes the enzyme's shape and can alter its affinity for substrate or its overall activity. In this graph, the x-axis is substrate concentration and the y-axis is reaction rate. If each binding site acts independently, we get a hyperbolic curve. That's the one in blue. The reaction rate rises quickly and then plateaus as the enzyme becomes saturated. However, some enzymes have multiple subunits that quote-unquote communicate with each other. This is called cooperative binding. In this case, we get a sigmoidal curve, which is that green S shape there, because the binding of the first substrate makes it easier for the next one to bind. A classic example is hemoglobin, which has 4 binding sites, so the binding of one oxygen increases affinity at the others. So, if you see a sigmoidal curve on a graph, that's a giveaway for cooperativity. Something called allosteric effectors can shift these curves. Activators make binding easier, shifting the curve to the left. For example, ADP can act as an activator in metabolic pathways, signaling that cells need more energy and speeds things up. Notice how it takes less substrate to reach a higher reaction rate. On the other hand, inhibitors make binding harder, shifting the curve to the right. A classic example is ATP acting as an allosteric inhibitor in pathways like glycolysis, signaling that the cell has enough energy and slows the pathway down. In this case, it takes more substrate to reach the same reaction rate. If this helps simplify allosteric regulation and cooperativity, drop a like and subscribe, and I hope to see you in the next video. Thanks.